So in this video, we're going to learn the complexity that is the alternative pathway of complement activation. So this is one of the mechanisms by which complement can get activated by a pathogen. So I've drawn here a pathogen. This is, let's say, for example, a typical bacteria. And bacteria has, let's say, gotten into the body through a cut. There's some tissue damage, and now there's bacteria in the body. And this could be occurring in, the damaged tissue, um, interstitial that's happening outside of a cell, outside of a human cell. So we're in fluids, we're in blood, we're in interstitial fluid, we're in lymph, for example. And that's where you find this protein called C3. It's a complement protein. It's a very abundant complement protein. And this is going to initiate the alternative pathway of complement activation. So let's see what's going to happen here. So the complement protein C3 um, can hydrolyze to a slightly different form. So what does that mean? That means that some of the reactive parts of this protein, C3, um, undergo hydrolysis such that this protein changes slightly its shape and forms a version that we're going to call IC3. So when C3 protein is in the vicinity of pathogens, like a bacteria, for example, the environment where pathogens like bacteria are replicating, the molecular structures on the surface of bacteria enhance the conversion of the C3 protein into its IC3 version. So hydrolysis occurs on the C3 protein, um, putting it into this different, slightly different version called IC3. And this occurs at a higher rate in the presence of bacteria. So something about the molecular structures and in molecular microenvironment of bacteria triggers C3 to be converted to IC3. Okay, well, we've got, here's another protein, factor B, another complement protein. It's called factor B. Now, factor B also present in the blood or the lymph or the interstitial fluid. Remember, all these complement proteins are made all the time. And what factor B is going to do is it's going to bind IC3. So factor B binds IC3. Okay, it doesn't bind C3, binds IC3. Interesting. Oh, there's another protein coming along, another complement protein, factor D. Factor D is a protease. Okay, we know what proteases are. We've talked about them in the previous video. What do proteases do? Proteases cleave proteins. So I'm going to introduce this concept to you now that factor D can cleave factor B only when factor B is associated with IC3. So you'll see there, factor D didn't attack factor B on its own. It only attacked factor B when factor B was bound to IC3. So factor D is a protease. Its substrate is factor B when factor B is bound to IC3. Okay. Well, now what happened to factor B? What did I draw there? Well, factor D is a protease. It cleaved factor B, broke a peptide bond, and separated two parts of this factor B protein. And we're going to call these two parts BB and BA. So BB is big B, little b. BA is big B, little a. The part that remains interacting with IC3 is the BB part. Factor BA, we're not going to talk about that. Don't worry about factor BA. Factor BB. So far so good. So factor D cleaves factor B into BA and BB, and BB stays associated with IC3. Well, this complex of proteins of IC3 and BB, it is now a protease. Wait, was it a protease before? No, no. Remember this term zymogen, where something might be in an inactive form and now it's an active form? Well, before, this was not a protease. When IC3 and B were together, not a protease. Now that factor B is cleaved, now it's a protease. So IC3, when it is bound to BB, is a protease that we're going to give it a name. we're going to call it the soluble C3 convertase. So we just formed a protease, and we formed it by using a protease. So we've got like what we call these a cascade. So factor D kicked off the cascade, and now we've got another protease called the soluble C3 protease. I'm sorry, soluble C3 convertase. And that is made of two proteins, IC3 and BB. So far, so good? Okay. So, okay, this is a protease. It's got to cleave something. Well, look at that name, soluble C3 convertase. Soluble, floating around. It's a C3 convertase. What do you think a C3 convertase is going to cleave? Well, it's in the name, C3. So, now where did C3 come from? Well, there's C3 all around, right? There's plenty of C3 in the blood, in the interstitial fluid, in the lymph. So IC3 and BB, which is the soluble C3 convertase, it will find a C3 protein that's in the area, and it's going to do what to it? It's going to cleave it. It's a protease, cleaves it. What is going to happen when you cleave C3 protein? Well, it's going to make two smaller proteins, C3A and C3B. So all we did was we activated a protease. We used that protease to cleave a protein. So the substrate of the soluble C3 convertase is C3. The products are C3A and C3B. Okay, so far so good. C3A, we're going to talk about in a later video. It does something, but not involved right now in what's about to happen on the surface of the pathogen. When C3B is formed, it exposes a very reactive thiol ester bond. So if you recall back from organic chemistry, there's a sulfur in here and a carbon with a double bonded oxygen. This is a highly reactive functional group. And what this reactive group can do is it can react with other molecules, such as one on the surface of a pathogen. So this C3b protein is very active, and it can often attach itself covalently to the surface of a pathogen. So what we have now done here is we have started this little reaction that caused C3bs to form, and C3bs will react covalently with the molecules on the pathogen surface. So this is the process called complement fixation we've referred to in the previous video. Complement is being attached covalently to the surface of the pathogen. So the pathogen is recognized initially by the C3 protein converting into IC3, and the result here is complement fixation. Interesting. All right, well, what happens when you have C3bs attached to the surface of the pathogen? This is a good thing to mark a pathogen for destruction. So attaching C3bs, which is, again, complement fixation, is a good thing to help get rid of the pathogen. So let's see what that does, attaching C3bs to a pathogen. Oh, look, it's our friend factor B. All right, it's another complement protein, factor B. There's plenty of it. Factor B is going to come over here. It's going to interact with C3b. Now, wait a minute. Didn't we have factor B interacting before? Yeah, we did. We had it interacting with IC3. Now, guess what? Factor B has multiple things to do. It can also interact with C3b. Okay, that's interesting. Oh, look, it's our friend factor D. If you recall from the last slide, what is factor D? It is a protease. Well, we saw in the last slide that factor D attacked factor B, but only under certain conditions. Well, guess what? This is also a condition that factor D, which is a protease, can bind and cleave factor B into BA and BB. When that occurs, you now have, again, the C3b protein that is covalently attached to the pathogen surface, and you've got factor B cleaved into factor BB and BA. BA floats away. Don't worry about it. Not important right now. So now we have BB and C3b. What is that? That is another protease. Before, if you recall, we made a soluble C3 convertase, and it's floating around near the surface of a pathogen. Now we have a C3 convertase attached to the surface of the pathogen. This protease has a name called the alternative C3 convertase. So the alternative C3 convertase is consists of the protein C3b, which is fixed to the surface of the pathogen covalently, and the protein BB, right? Which is a breakdown product of factor B. Where'd that come from? Factor D cleaved to factor B. Only the presence of C3b in this instance. Okay, so now we have another C3 convertase. Do you remember what C3 convertases do? This is our second C3 convertase. What do C3 convertases do? Guess what? They take factor three, they take C3s and they cleave them into C3A and C3B. Now, if you recall, what happens when C3 is cleaved to C3A and C3B? C3A we'll talk about later, but the C3Bs, they have that very reactive thiol group. And what does that group like to do? It likes to react with other molecules. And in this instance here, it's reacting to molecules on the surface of the bacteria and attaching. That's more complement fixation. So the end result of the alternative pathway of complement being activated is that many C3Bs become affixed to the surface of the pathogen. How? Well, C3s got cleaved first from the soluble C3 convertase and then forming the alternative C3 convertase amplifies the amount of C3Bs attached to the pathogen. So this whole process results in many C3Bs covering the surface of the pathogen. What's that going to do? Well, we'll cover those effector functions of complement in a later video. But the point of these videos is for you to understand the proteins involved in recognizing a pathogen using the alternative pathway and the resulting convertases, C3 convertases that trigger the complement fixation onto the surface of pathogens.